Very honored to be here to present the work that is done in our team in using deep learning to model ecological communities. But while the caffeine is kicking in, I will give you a review of some basics. So what do we mean by an ecological community? A community in ecology is an assemblage of populations of two or more species that occupy the same geographical area at a particular time. In these communities, species are they do not exist independently. Instead, they interact in various ways, either indirectly, through the habitat, for instance, or directly, by interacting directly, such as by feeding on one another. And these interactions are typically depicted using the formalism of ecological, using the formalism of graphs as ecological networks. What we mean by community data is the exhaustive survey of which species are present or absent at a given location within a given time window, and is typically surveyed in multiple locations and sometimes at multiple observation times using various sampling protocols, as you can see in this AI-generated image. For instance, soil microbial communities would be sampled using environmental DNA, for instance, whereas bird communities can be sampled, for instance, using passive acoustic monitoring. When we talk about community models in this presentation, we refer to statistical models that relate community data to environmental variables. And this includes species distribution models and their multi-species extensions that model species composition. Sorry, and it includes also microecological models, which model functional or taxonomic or phylogenetic diversity indicators. The goal of community models is a double quest to improve our understanding about the factors and the processes that shape the distribution of species, and also to be able to do predictions in space and also in time under various hypothetical scenarios, as you've seen earlier with Damaris. And all that to support conservation and restoration actions. So in practice, how do we model multiple species together? The most naive approach to do that is to simply stack individual SDMs, one for each species, and to predict each species individually and then simply stack their probabilities. And one of the major inconvenience of this stacked approach is that it overlooks the impact of ecological interactions. And there is increasing literature that shows that ecological interactions actually influence the distribution of species, so we have to account for that. And in fact, not only direct interactions, but also indirect interactions, and also the shared response to the environment and biotic drivers, it creates statistical dependencies between the distribution of species that we refer to throughout this presentation as species associations. So to account for these associations, there has been development of joint species distribution models, as you've seen with Florian this morning. But also, you can use your favorite machine learning models that are implemented in SDMs, and most of these algorithms, they have multi-output extensions that you can use to model multiple species together. And actually, DeepSDM are a particular example of that, in the sense that DeepSDM are simply deep neural networks that take as input environmental covariates and predict multiple species probabilities. As you've seen, and I guess it's the point of today, is that DeepSDMs are a promising avenue. They have been shown to provide high predictive performances. They can adapt to high resolution data such as satellite imagery. They are scalable for large-scale predictions. And now we have tools that can be used to test and evaluate these approaches. So a typical architecture of a DeepSDM contains, you can summarize that into two components. The first component is what we refer to as the environmental encoder, which is a neural network that transforms the environmental data into a vector, what we refer to as embeddings, which represents a new representation of the environment that is more relevant to what we are trying to predict. And this is, in fact, similar to when you're using principal component analysis to reduce the dimension of your environmental vector, except this is fancier, in the sense that it can account for complex environmental data. For instance, one of the advantages of this module is that you can adapt this feature extraction to the type of data that you're using. For instance, for image data, you could use convolutional neural networks, CNNs, or vision transformers, or some fancy hybrids of these two. But you can also combine different modalities, as we've seen this morning. The second component of this DeepSDM architecture is the classification hat, which takes these embeddings and simply predicts the probabilities of species. And it is itself another neural network, typically fully connected, but not strictly. And as we've seen this morning, its strength lies in the fact that it is shared across species. And so there's a sharing of statistical strengths between species, which tends to benefit rare taxa. And it acts also as a regularization across species. So for my part, I will talk to you about ways by which we account for species association in DeepSDMs. And then my colleague Selene will present Earth observation foundation models and their application to model communities. So let's start with associations. Here, the idea is to build on this baseline DeepSDM architecture and to add components that capture different types of associations and to see how we can implement that and also how can we interpret that. So the first kind of association that we can model is the association through the shared environment. And this is an implicit association in the sense that we're not explicitly modeling it in the model, but it emerges from the way the model is formulated. And this is already accounted for in the baseline DeepSDM architecture. Here we assume, by doing that, we assume that species are independent conditional to the environment, and that all the factors that explain or drive the dependencies between species are actually in the variables that we are already providing to the model. We have proxies for these variables in the data. For instance, if we are not measuring light, but we are measuring vegetation structure, then we can use it as a proxy for light conditions in forests, for instance. To illustrate this, I will use a case study of plant communities in the French Alps, based on a study by my colleague Yu-Chin Hu, where we modeled multiple species as a function of a combination of tabular data that includes climate, soil, and topography, and images based on infrared color orthophoto imagery. We used a multimodal architecture that combines an MLP for the tabular data and a CNN for the images. And then we combined these embeddings. And once the model was trained on species distribution, we extracted the embeddings, reduced their dimension, and then visualized them in a 3D space using UMAP. And then we projected, and then we visualize here on the far right the clusters that have been computed on the original embeddings. And here I show for 3 clusters example of images that have been assigned to this cluster. And for instance, in the far, in the top left, you have alpine images of alpine meadows that are sparsely vegetated, typically occurring at the edge of screes or snow melting edges. On the top right, you have light oak forests, and these are, and for each of these clusters, you can see here species that are strongly associated to these clusters, and what we see here is the species that are associated to the same habitat. So this is an example of species that share the same habitat preferences, so they are positively associated through the habitat type. Now, so far, we have assumed that we are accounting for all the factors that explain why species depend on one another. So, in general, we know that we do not measure all the relevant factors. And in fact, in some ecosystems, such as the soil ecosystem, we don't measure the most important variables that actually explain why species interact, how species interact with one another. And so in this case, what we do, we augment the previous architecture, and we add the notion of latent variables. And so we use a similar formulation as latent variable joint species distribution models, where we assume that the response of the species is a total of its response to the environment, plus its response to these hidden or unobserved factors. With the difference in that the observed environmental variable can be computed using from images, from tables, and from any flexible input. And so one of the nice things about this formulation is that you can reconstruct a species association matrix using the species latent factor loadings. That is the parameters of species response to these hidden variables. Now the challenge here is how do we estimate these latent factors and the other parameters, given that we don't observe these latent variables. And so to do that, we use a combination of our classical DeepSDM, and we combine that with a variational autoencoder to estimate the latent factors. And the idea here is that these latent factors that are not observed, they need to be sampled from a posterior distribution that is estimated on the data. And we cannot integrate posterior Bayesian sampling with deep learning training. It's too computationally expensive. So to do that, what we do is that we approximate the posterior with an approximate distribution, a variational distribution, Q here, and we use a neural network to predict for each sample the parameters of this distribution. Here, since it's a Gaussian distribution, we predict the mean and the variance of this Gaussian distribution of the latent factors. We sample from this distribution, and then we obtain latent factors. Then we combine these latent factors with environmental embeddings that have been extracted using a DNN. And the combination of these two factors is the input to species-specific neural networks that predicts species independently. So we apply this approach to earthworm communities that have been sampled by Marcel Boucher in all over metropolitan France, and described in his book that was translated into a database by Michael Ed from INRAE. So in practice, we simply translated the architecture that I showed earlier. We modeled these earthworm communities as a function of both in situ field measures and variables extracted from GIS databases. And here, in this context, the latent factor can represent, for instance, the soil matrix, the microbial activity, or, for instance, the vegetation that was not sufficiently well described in this data. And I am just going to skip all the results and just show you the species association network that we obtained. Here, we modeled 77 species, but only 31 out of them were involved in associations. We obtained a network that has a core periphery structure, where the hubs represent dominant species of earthworms in France. And what we see, I didn't put any pictures because for me they all look the same. I'm not expert on earthworms. Here we're one eminent example that we could interpret with Michael. We had these two species of Poectodea caliginosa, caliginosa and meridionalis, these two. They are subspecies of the same species. The first one on the top occurs in all the territory, even in the south, but in areas in the south where we found the meridionalis, we never found the other one. So there is a strong negative association that is not explained by the environment, and it is captured here. And on the right, we have a ternary positive association between three species that tend to co-occur generally together in northern France. Okay. So, so far I've shown you implicit dependencies. Now we can also build on our ecological knowledge to enforce explicit constraints in the models. Now, one type of constraint that you can enforce is a macroecological constraint. Here, as Damaris was showing earlier, species distribution models can predict species communities, but they are notoriously bad at predicting community-level indices. On the other hand, macroecological models are good at predicting community-level indices because they optimize for these indices. However, they cannot inherently predict species composition. So here the idea is to use macroecological models to constrain predictions from species distribution models in order to achieve two objectives. The first one is to improve the prediction of observed community composition, and the other one is also to refine the existing predictions by the macroecological models. And here the kind of association that we are leveraging is the association between a species and whole assemblages. So in practice, what we do is that we use functional indices that have been predicted, for instance, with macroecological models. And we use predictions of SDMs that have been derived in whatever SDM framework you want. And we use a neural network that combines these two inputs and predicts a new composition of species. However, this composition, when trained, it is optimized on both the community composition, the observed community composition, but also on the emerging community indices that are computed from this predicted composition need also to be as close as possible from the observed community level indices. So we applied this to a data set of plant species communities in the Alps. Here, as community level indices, we use species richness, and we also use community mean and standard deviation of 3 traits: specific leaf area, leaf nitrogen content, and plant height. And what we did was we evaluated this framework that we call Camellia. We evaluated using, first we trained it on the training data set, and then we evaluated using different variants of the community level indices. In the first one, we use the ops in green represents a situation where we use observed community level indices and we want to predict community composition. And the second one is by using the estimated estimated indices from microecological models. And the third one is using the observed observations but adding noise. And the idea here is that for some community level indices, we can have an estimator for these indices from remote sensing, for instance, such as plant height. So the idea is to at least simulate this pattern. And so what we found is that this Camellia framework improves the scores of species prediction using both observed or noisy community level indices. However, when we compare, and we compare this to stacked SDM, because this is our baseline, when we compare however, when we use macroecological model estimated indices, we have a trade-off between insensitivity and specificity between Camellia and the stacked SDM. Okay? So the second type of constraint that we can enforce in our DeepSDM models is to use the knowledge that we have about ecological interactions. Now, maybe you didn't know that, but there's a growing wealth of species interaction data that are both derived from expert knowledge or from experiments and observations, or sometimes even from their footprint on co-occurrence patterns. And this interaction data is typically shared in various interaction databases, such as Globi or Mangal. And the idea here is to leverage this interaction network to better predict the distribution of species. And this is based on the work of Giovanni Poggiato. Here, the components that are needed for this model is environmental layers, a metaweb. What we mean by a metaweb, it means a global interaction network between species that is not spatially explicit, that just says these species tend to interact with one another. With the constraint that this network has to be directed and acyclic. You will see later why. And we also need, obviously, observation data, community data. So the assumptions here is the metaweb has to be directed and acyclic. It has to be stable in space and time. And we have to choose a direction. So we either say that we have a top-down control. It means that it's the predators that influence the prey. Or we say that we have a bottom-up control in the meaning that it's the predators that depend on the prey and not the other way around. And so under these 3 assumptions and using the causal Markov condition, we can obtain this property, which is that given its prey, a species is conditionally independent from the prey of its prey. So basically, if you want to model a given species, let's say the rabbit here, let's say the owl. If you want to predict the owl, we only need to have information about the environment and its resources. We don't need to have information about the resources of its resources. Maybe the better example is the wolf, where we simply need to have a model that combines, that is a function of abiotic environmental conditions and the otter and the deer. So basically, in this situation, the problem that we have will translate into S-independent local models, where S is the number of species, and each species can be fitted independently in parallel. This is during training. Now, the challenge is when we want to predict in areas where we don't observe the resources. In this case, we have to start from the bottom if we are doing bottom-up control, or start from the top if we're doing top-down control. So what we do is, first we do the basal species, and what you mean by basal species is species that only depend on the environment, that don't have resources, at least in our pool of species. And then once we predict these species, we predict their consumers and so on. And in general, when you have a trophic chain, you can get this order automatically. But when you have a complex food web, a complex interaction network, you need a topological sorting algorithm to obtain this order. And this is actually fairly easy to do. So in summary, building on this baseline DeepSDM architecture, we can add different components to incorporate either implicit dependencies. These are associations between species that emerge from the model structure. For instance, associations that are due to an environment that is measured, or to some factors that are not measured, in the second case. Or we can also enforce explicit constraints. And here I've just shown examples using macroecological or non-interaction networks, but we can think about many different constraints that can be used to guide the models or to act as a regularization towards better predictions. So take-home messages from this part. Deep learning, as I wanted to show you, that deep learning contributes to community modeling not only for predictive purposes, but it can also be guided with ecological knowledge. And for me, personally, I think the biggest strength of deep learning, in addition to this multimodal representation learning, is this capability to be flexible enough in the way we formulate the models, such that we incorporate ecological knowledge. And as I showed you with the variational autoencoder example, deep learning can also serve as an inference tool. When we want to do, it can be used, for instance, for variational inference when we don't want to have a heavy, computationally heavy Bayesian inference. Now, before I leave the floor to my colleague Selene, community data are increasingly available, but they still remain small in size in comparison to typical deep learning data needs. So one of the best direction for me to follow is to, instead of training these deep learning models from scratch for each application, to leverage pre-trained deep learning models we refer to as foundation models, to reduce the cost of this DeepSDM training and to reuse models across multiple applications. So I leave the floor for Selene.
Hello, everybody. I will continue with the second part of this presentation, and is related to Earth observation foundation models for biodiversity modeling. So let's start with some definition. What are E of F models? Well, they are deep learning architectures pre-trained on large multimodal EO datasets like hyperspectral data, multispectral data, digital elevation models. In some cases, it's text. So all this data comes from different Earth observation platforms. And so how Earth observation foundation models works? They learn general habitat and landscape features across spatial and spectral dimensions in a self-supervised way. So when we talk about self-supervised way, actually we are presenting three main categories, which is contrastive, predictive, and generative. And in the case of generative, we will find an example like masked autoencoders. And the main goal of this strategy actually is to reconstruct the missing parts of an image that has been hidden randomly. So these EOF models, are they useful? Yes, they can be fine-tuned for specific environmental or ecological tasks, for example, for object detection, for change detection, for like to monitoring wildfires, floods, terrain, etc. So here I present you some Earth observation foundation models. Depending on the architecture, we can see some based in convolutional neural networks like SECO-ECO, And the recent ones are based in vision transformers. Here we can see DOFA, which here the strategy is masked image modeling and it uses input data from different sources. We have also PRITV that was developed by IBM and NASA, and they use multispectral data from Sentinel-2, and this is actually in 3 time steps, and in total they use 18 layers. And we have more examples like Copernicus FM that use a unified data set, and this unified data set actually considers all the Sentinels 1, 2, 3, 5, and the digital elevation model. And we can see other models like GeoLandBind and TerraMind and others that we can see in the literature. But the main focus of this second part is actually how we can integrate these EOF models for biodiversity modeling. So we performed several tasks, and at regional scale and at European scale. So let's focus first in regional scale. And here the region was the French Alps. We extracted data related to environmental data, remote sensing biodiversity like phenology, for example. And we also considered orthophotos, RGB and infrared orthophotos. We put them all together to try to predict soil biodiversity. So let's enter in more detail here. Our data set for this case, for the regional evaluation, we use it, actually it's a small data set of 1,000 soil samples around. And we used environmental DNA. From here, we computed the soil trophic group abundances. And we also included climate data, soil properties, phenology, landscape composition, and orthophotos in high quality, like 20 centimeters of resolution. So, to be more precise, the task here, the goal of this task was to predict the abundance, the relative abundance of 51 soil trophic groups in the French Alps. And someone can make the question: are you trying to predict below ground biodiversity using aerial images? And yes, challenge accepted, because the main idea is that we have data, field data that is high quality, but also we are looking for another approach, and one based on aerial imagery. So, but we found some challenges, like this first one, that there is a taxonomic and technical limitation in species identification. And how do we overcome this problem? Well, we used environmental DNA, metabarcoding, to estimate soil trophic group abundances. Another challenge was the lack of large and high-resolution environmental data sets, because as I mentioned before, our data set is small, 1,000 samples. So we were thinking how we can extract rich features from the images, for example, if it is possible from tabular data. So for this reason, we thought, okay, Earth Observation Foundation models can give us these rich features, and we can try it in our models, in our regression models, to predict the abundance of these trophic groups. And another challenge is the difficulty of integrating diverse data types for soil prediction, because we have tabular data, for example, climate data, soil characteristics, and how we can integrate it with images. We need to process these images in some way. So with Earth Observation Foundation models, we will do that, and we will construct at the end some regressors to predict abundance, to predict soil biodiversity. So here is part of the methodology that we leverage the EOF models. We took the orthophotos that we have, and we inputted them to two types of models. We selected 2 models, SAT-DINO and DOFA. And actually we choose these models because, as you can see in the characteristics, SAT-DINO was trained in forest, mountainous, and tree biodiversity. And in the case of DOFA, it was trained in different type of data, radar, multispectral. And this is beneficial for us because we use it for RGB orthophotos and also infrared orthophotos. So once we input all these images to these models, we extracted embeddings, the features, but this embeddings belongs to the penultimate layer. And with these embeddings and with all the data that I described before, the abundance, the climate, etc., we organized all this information in 4 data configurations. So first, what we are going to predict is, as you can see, the soil trophic group abundance. And then we have these 4 data configurations. The first is using or is based in coarse gridded climate and soil data, and we also added phenology and landscape. In the second case, we used high quality for climate and soil, and this data comes from Orchon. This is field data. And we have also the third case. Here, the predictors are just embeddings. With embeddings, we're trying to predict the abundance of these trophic groups. And in the fourth case, we are considering the embeddings plus the coarse gridded data. It means with all the data that we consider in the first data configuration. Why? Because we wanted to test different approach, if embeddings can give us a good result or some similar result to when we use high quality data. And as you can see, we tested 3 machine learning techniques, well-known, LightGBM, random forest, a multi-layer perceptron. And to avoid data leakage, we performed altitude stratified cross-validation. Then we obtained the predictions of the abundance, and we elaborated some maps. So here we have the results for soil biodiversity prediction. As you can see at first sight, there is not a big difference between each data configuration. And there is a slight difference by machine learning technique. But the big difference, the remarkable difference actually, is between the biological category. Bacteria, fungi, and protists showed better results than, for example, oligochaete and insect. And of course, there is something behind this, because oligochaete and insects are hard to sample. And bacteria, fungi, and protists are abundant in this case. So if we want to check more in detail if there is or no difference between data configurations, here, for example, we can see that in purple, the cases 1 and 2, which used or were based in tabular data, presented better results than the cases 3 and 4 that considered embeddings. So, okay, tabular data is good. Yes, high quality data is good. Yes, that's the second case. But what is happening with embeddings? Why the embeddings are not showing better results? So we were trying to dig a bit more to understand what was happening with these embeddings. And we elaborated a PCA projection. And you can see here the visualization of these 2 components that capture the majority of the variance. And the embeddings that we are projecting here are from SAT-Dino, the model that shows us better results. So we can see that there is certain differentiation in the land cover, in the dominant land cover. But if we check, for example, in the side with gray, yellow, and red, so we can see that the grassland shrubs might have inherently similar visual features, making them harder to separate in the embedding space. So for soil, at least, we need fine scale features that, in this case, embeddings can't give us. But at least we can see that we can obtain more or less good results. So so far, the take-home message of this study are: there are group-specific predictions challenge across trophy groups. Maybe there is more information or specific information that we have to add for each biological category. Tabular data outperforms embeddings. Yes, the embeddings capture partial information, and the embeddings can offer actually an alternative where in situ data is scarce. So we validated that. And also we performed other 2 downstream tasks, but in this case was to European scale. And here we focus on Europe. We extracted again environmental data, remote sensing biodiversity like phenology. And instead of using orthophotos of high resolution from IGN, we used multispectral data from Sentinel-2 and radar data from Sentinel-1. And again, we extracted the embeddings. We put all of them together, and we performed the prediction of soil biodiversity. In this case, a big scale, European scale, and we also predicted the functions of them. And we also performed the classification of UNI's habitats. So I will go through each task in detail. Soil biodiversity prediction, in this case, we worked with more or less 60,000 to 1,000 soil samples. We have chemical properties here, soil functions, microbial activity, soil trophic groups also. And we have climate, terrain, soil texture, and in this case, we used just multispectral imagery from Sentinel-2. In the case of habitat classifications, we worked with more or less 700,000 vegetation plots, UNIS level 3 and 249 habitat classes. And here the resolution was 100 meters. And some of the variables, well, actually are similar: climate, terrain, soil, geology. Well, we added some other variables also here. And the difference with the soil is that we added a radar imagery. So the results for the first part of soil biodiversity, but a European scale in this case, we can see that at least in the y-axis we can see the target groups: bacteria, fungi, metazoa, and we have also chemical, enzymes, and microbial, physical. And we can see that for bacteria, when we added this multispectral imagery, actually the model underperformed. And for fungi, there was no change. But for the rest of them, there was an improvement. And we contrast more actually for the part of chemical, enzymes, microbial and physical functions. So there is some improvement, but just for certain target groups. And here we can see more in detail for each trophic group and each function. We can see that for which ones actually there are a big improvement in this side, going this direction, and the models that underperformed actually in the left side. In the case of habitat classification, here we have. We did predictions for each habitat, and we can see that in orange we have the baseline. And when we added environmental data, like climate data, for example, in blue, we can see that we can obtain a better, definitely, result for prediction, for classification of habitat. And if we add remote sensing products like phenology, we can also see that there is some improvement in most of the cases. While we add in multispectral data in red, for most of the habitats we can see an improvement, exception for shrubland, S. And if we add radar data also, there is improvement in all the habitats. So here actually we are showing the baseline with the models that I mentioned previously, but in this case we are testing different data sets to see the performance of our models, and we compare also different Earth Observation Foundation models. In general, we can see that, okay, if we add environmental data, remote sensing characteristics, we obtain good results, yes, but we don't see big improvement for some Earth Observation Foundation models. Actually, they are similar. They are quite similar. So for these two tasks, some take-home messages is that EOF models embeddings from multispectral and radar imagery, they provide complementary information to both remote sensing biodiversity products and environmental GIS databases. At fine scale modeling, we can see that actually multispectral and radar embeddings contribute highly to biodiversity models. And so in general, about this second part of the presentation, we can say that actually EOF models offer valuable habitat embeddings for biodiversity modeling, especially when there is a scarce of data. EOF models complement existing remote sensing products such as phenology and land cover data. And their contribution actually varies by spatial scale and taxonomy group. And for this reason, our future work will focus on broader testing across diverse taxa and modeling tasks. And as AI for conservation scientists, the use of pre-trained EOF models allow us to reduce the computational cost and thus the carbon footprint of our research activities also, if we can reduce them. We would like to thank to our team, LECA, our collaborators, providers, stakeholders, and founders. And this is our team. I love the team. And that's all. Thank you for your attention. And do you have questions?
Thank you. Very nice talk. We may have many questions. Who's first?
Okay, so one question about the embeddings and the relatively low performance of these foundation models. So, I mean, I know you're not supposed to do this with a foundation model, but there would be, of course, the option to fine-tune the model to get a kind of semi-supervised. Did you try to, if it would improve the performance? Because the question is, in the self-supervised learning, does it not learn the right features, or is there just no signal in this data? So I was wondering if you try to fine-tune the models also to go to a supervised approach.
It's a great question. Actually, so we're trying to test the fine-tuning also different model fusion architectures. But here we wanted to see if we have a data scarce setting where we cannot afford to do a lot of fine-tuning either. Can we still do, how well can we do just with the baseline embeddings? How well can we get? This is in a way a baseline. And then obviously I think it's a good way to go to try to see if fine-tuning would be helpful in this case.
Yes, I had a question on another topic that you mentioned, Sarah, which was integrating the knowledge of interactions to predict species distribution. So you mentioned an approach, very interesting, that is, however, constrained by a directed acyclic graph. Yeah. I was wondering if there exist approaches for more general interaction graphs, like mutualistic interactions, the plant pollinators networks, or more complex stuff.
Yeah, obviously the easiest one is using DAG. That's the one I presented. But I think in mutualistic or competition graphs, first you have to have a knowledge about who's competing with who, and usually, at least for competition, it's usually dynamic, and you have to have some dynamic measures of the resource that they are competing for. I guess they were targeted, they were the main target of JSDMs, estimating this potential competition in associations, but there are many publications that show that it's not, the negative correlation or associations are not necessarily synonymous to competition. For mutualism, there are so many types of mutualism that I think you have to think in a case-by-case basis, and maybe sometimes you have actually mutualism through the environment, and you're already measuring it. So I think it's difficult to just say there is a global approach that works for all types of networks. You have to think on a case-by-case basis. At least that's my opinion.
We have time for a last one. Yes.
I was wondering whether you investigated the idea of building an end-to-end model, which could use or predict both macroecological features and species-specific ones.
Yeah, we did not try that yet, but it's in the plans. I actually have a hidden slide to show that, if you'll allow me. Sorry, yeah, this is it. So here, is this what you mean? So from the environment, we predict directly both the community composition and the community level. And this is an ongoing topic, but we haven't started yet. So if you're interested to collaborate, feel free to reach out later.
Cool.
Thank you. So next speakers.